So this is a video that's going to go through the process that we just talked about in the webinar, the process of DNA isolation from plant tissue. There's a number of methods that are both physical and chemical, organic and inorganic, that can be used to extract DNA from plant tissue. But then for this video, we're going to go through the chemical organic DNA extraction process that uses phenol chloroform for DNA extraction. So for that, we'll need our reagents, which are listed as follows. So, as we discussed earlier on in the webinar, we talked about the reagents that we need for the process of DNA isolation from plant tissue. We need these for the specific processes that we talked about: cell lysis, digestion of protein, precipitation of DNA, and then finally washing the DNA. We have here our isoamyl alcohol, we have our chloroform, our isopropyl alcohol, and then here we have the CTEP buffer, and finally the DNA wash solution. So we'll use these for the specific process that we are going to go through the video, and please try to pay attention so that you can use that information for the activity that's to follow. It's very important that you try to pay attention to what's being done in the video, because that's the whole essence of making a video about this, so that you actually know each and every step of the process, even though you are not here physically present. So the first thing you want to do is make sure that you weigh enough amount of plant tissue that you're going to use. Here we're going to use leaves from common reed, which is Phragmites, that we're working with here in the lab. You can see the balance that we use, and I'm going to weigh approximately 7 grams, yeah? And then I'll use 1 gram for the experiment. So what you want to go ahead and do now is grind your leaf tissue in a chilled mortar with liquid nitrogen. So what the liquid nitrogen does is freeze the plant cell, and when that happens, the plant cell is easier to break mechanically, and it also deactivates your natural enzymes like the DNases and the nuclease, so that your DNA remains intact, it's not degraded. So you want to make sure that each and every leaf that you have is frozen, and that makes the breaking down of the cell easier. You can follow, as you can see in the video. Make sure that that's done properly, because this is the very important step. It makes it exposes it helps expose your DNA so that it can be easily isolated. Keep on adding liquid nitrogen as needed, so that you can completely have a pulverized sample. So make sure that you continue grinding and grinding until you have a flour-like sample. So you can see in this video, I'm continuing with the grinding until I have that type of sample, and that will help to assure that my process is efficient. This is the almost final sample after grinding. This is what it needs to look like. So continuing, I'm here making that solution of chloroform and isoamyl alcohol at 24:1 part. It has 24 parts of chloroform and 1 part of isoamyl alcohol. So you can see here I'm measuring the amount of each and making sure that I have the precise amounts for a precise outcome. So you can see this is that solution that will help us with the digestion of the proteins before we precipitate our DNA. So follow through and see how simple and easy it is, and that will help in proceeding to the next step. So of that sample that we have ground, we are going to go ahead and measure just 1 gram of frozen tissue, and then we'll place that tissue into a 15 milliliter plastic cap centrifuge tube, and then add 5 milliliters of preheated CTEB isolation buffer. This CTEB isolation buffer, like we discussed in the webinar, is going to be preheated to 60 degrees Celsius. And then after that, we can go ahead and incubate it for 30 minutes. So you can see here I'm doing the measurements for the 1 gram specifically that I need, and then I'll go ahead and add that solution of CTEB isolation buffer, and then repeat and again incubate it for 30 minutes in 60 degrees Celsius. That's in a hot water bath. So you can see the process is simple and straightforward. And here, like I said, that's when I went on and added that solution of CTEB buffer. So repeating again, following the protocol that I sent in the webinar, here it's just adding 5 mils of preheated CTEB isolation buffer, just 5 mils, and then incubate for 30 minutes at 60 degrees Celsius in the water bath. So I'm going ahead and incubating my 2 tubes in the water bath. One with my tissue and my CTEB, and the other one with my CTEB. As I wait for that 30-minute incubation period, I'm going to go ahead and prepare this tube with that mirror cloth that we talked about in the webinar, so that I can separate my solutions after the centrifuging. So you can see here I have it ready and prepared, and I'm going to go ahead now after the 30-minute, this is after the 30-minute incubation period, and I'm going to go ahead and add 5 mils of that chloroform and isoamyl alcohol solution that we made earlier on, and then after that I will centrifuge for 15 minutes at 3,500 rotations per minute. I hope you're following through. You can see how simple and easy, straightforward this process is, and how each and every step is done in the lab. So make sure that after you add your 5 mils of that solution, you go ahead and shake vigorously so that you can mix, and then you should also periodically open the lid so that you release the pressure in that tube. So this is a solution that you have after, and make sure that after that you go ahead and centrifuge it. So you can see in this next step, I'm going ahead and preparing the centrifuge machine. I'm going to set it up at 3,500 rotations per minute for 15 minutes, and after that we would have to separate our DNA aqueous solution, and then we can precipitate it using isopropyl alcohol after. So this is the basic process, and we are about to get to the end, and I hope you followed through and you've understood each and every step. Please ask questions in the email or via the next webinar if you have anything that you think you did not understand. As mentioned earlier on, are the conditions that we need for this centrifuge: 3,500 rotations per minute for 15 minutes, and then you can also see that the temperature is at 4 degrees Celsius. So we wait for those 15 minutes, and then here you can see we have our final product. So continuing, we take that top layer of solution, and we use that tube we prepared with a mirror cloth to extract it and pipette it into a clean tube, allowing it to stray through the mirror cloth, eliminating all the particles from our tissue, and this will typically yield us about 3.5 mils of aqueous solution. And with that aqueous solution, we are going to go ahead and add 2.5 mils of cold isopropyl alcohol to precipitate our DNA, and that will be probably one of our final steps. So you can see how this process is done, how you can pipette your top layer of liquid into or through that mirror cloth, and then you can have that aqueous solution that you can precipitate the DNA from. So make sure you are able to follow each and every step, you understand the process, and if you have any questions, you can ask via email, or you can use the next webinar session to have those questions answered. So you can see here we are almost done, and with that solution that we'll get after passing it through the mirror cloth, we are going to add this isopropyl alcohol that we talked about in the beginning, and then what our final product would look like is like this. So you can see that's our final DNA precipitated, the top layer that looks whitish in color. And I hope you enjoyed this video.